Hello, everybody. Um, I'm Ajasja Ljubetic. I'm a research assistant professor at the Department of Synthetic Biology and Immunology at the National Institute of Chemistry in Slovenia. Um, and we've got a very good web page that you're welcome to check out. So I will skip most of my CV. I will just share that I spent 3 wonderful years in the Baker Lab, like most of the people in this wonderful photo. So here I am. And I would also like to share that in 30 years, I would like to live in a world where protein robots can heal diseases, power synthetic cells, or perform computations or build smart materials. And that's why I think we should be designing protein machines and protein walkers. Natural protein walkers are amazing machines with huge potential. However, they're not stable outside of the cell, they're hard to modify and program, and the mechanism is not 100% understood. And this is where de novo designed protein machines can help because they can be hyper stable. You can even boil most de novo proteins. They have shapes that are easily programmable, and we can also identify minimal components needed for walking. So this is a hard problem. Nobody has built a completely autonomous de novo designed protein walker from scratch. However, we made some steps in this direction by designing a random protein walker. So we have a track that's made out of proteins. We have walkers that are de novo designed oligomers. And then for feet, we're using coiled coils of various lengths. And currently we have these flexible linkers here. So this is how we envision that the system looks like. But more importantly, we have experimental info. So this is a turf microscopy image. The fibers, the tracks are in green, and this is just a single foot here, this block. And you can see that a single foot just steps on the glass, doesn't interact with the track. However, if you put 5 feet on a walker scaffold, then you can see that the walkers walk along the fiber and you can track each individual molecule and get their diffusion properties. And we also have cryo-EM of the track and cryo-EM of the walkers. But of course, we don't see the feet because they're flexible and they get averaged out. And in the future, we would like to go from flexible linkers to well-defined rigid fusions that can give the track some asymmetry. And I'm very happy to announce we got an ERC Consolidator grant, Propel, which aims to design the first completely de novo designed autonomous walker, designed completely from scratch. So we'll focus on these rigid fusions, we'll design some anchors, and then we'll add power sources and demonstrate cargo transport. So now a few words about the National Institute of Chemistry. It's located in Slovenia, here in the middle of Europe. It's in Ljubljana, the capital city. Ljubljana is a beautiful city with a castle and an old river. It's been voted the European Green Capital in 2016. It's been voted among the top 25 small cities in 2020. We have universal healthcare, cheap childcare, and then cheap or free education. This is how the institute currently looks like. There's even a pool here in the background, although it's not part of the institute, and we're building a new building in 2027. So we have excellent equipment. We have a computer cluster with 64 GPUs and more on the way. So here's the cluster. We have a lab set up for high protein expression with like ACTAs and an autosampler. We have a single turf capable of measuring single molecules and also optical tweezers. This is a CTRAP Edge 350. And we also have a cryo-EM in-house, which is amazing. And we're also well connected into the protein design community, especially the Rosetta Commons, which is an amazing organization. So this is the current team, a postdoc and 3 PhD students. And I will be looking for at least a PhD and 1 postdoc in the summer for the ERC Propel. And the ideal postdoc should be interested in molecular machines and obsessed a little bit with proteins, have a biochemistry background and love at least Python, likes to work in a team, and would be happy to mentor students and share their knowledge. And of course, the skills you can learn in the lab are dry lab, de novo protein design, so how to use and run from RF diffusion to protein MPNN, Bindcraft, PulseGen. wet lab protein design, especially high throughput, not high throughput, but like medium throughput expression in bacteria, purification, stuff like that. Cryo-EM, single molecule turf microscopy, optical tweezers, and then of course you will have the opportunity to mentor students if you like. And you can contact me per email, but you can also, if you're curious about how things are here, you can also contact some of my group members and ask them how things are. And with that, I would ask if there's any other questions.
I mean, we had European RosettaCon at your place. This was awesome. It's really a wonderful city to be in and to live in. This is, I think, super exciting. Can you tell us what is the unique, thing that you're gonna attempt in your laboratory that sets you guys apart from everybody else working in this area? So can you tell us a little bit about what are your absolutely unique ideas that nobody else is pursuing?
Thank you. I don't want to go too much into the unique ideas because we haven't attempted them yet, right? But I think what sets us sort of apart is that we already have the random protein walker and we have all the technology to do the rigid fusions already. So I think we're well on the way to getting like an asymmetric Brownian ratchet type walker working. So that would sort of be the unique position currently.
Yeah, thank you. And Ljubljana, you just invited me to be there for the European RosettaCon, but this is an awesome place to
Thank
live
you.
and be. Thank you so
Thank
much
you.
for hosting us. But I had a very very good time with my kids there.
I'm very happy to hear that. Yeah, Ljubljana is great with kids. That's maybe also very thing to emphasize. It's a good place to raise kids.
All right, so my lab is at UMass Medical School. We are about an hour outside of Boston, driving wise. So the great thing about that is you don't have to pay quite as much, but you get to still be in the New England area if that's something you're interested in. It's a reasonable size, smaller city, still connected on the train. And we're in the biochemistry and molecular biotechnology department. So our lab's a little bit different than some of the other ones you might be hearing about. I did my PhD, graduated in 2012 with David. And then I switched to zebrafish. And my lab is trying to sort of bridge these two things. Now, you might see a zebrafish here on the screen. And the big picture of what we are trying to do is to develop treatments for neurodevelopmental disorders. So that's like the overarching goal of the lab. And you might think, why? What does Rosetta have to do with this? And where would we fit in? But we are interested in this process all the way from the protein level. And so we want to develop drugs. We want to make better tools. And we have ongoing research projects in the lab that are sort of working on that protein level, but it fits in with the entire lab, which goes all the way to animal behavior. And so I think our lab is a really ideal fit for somebody who wants to still make use of the skills that they learn, but go do something a little bit different or new in their postdoc and be able to sort of bridge those two areas. And not everybody works with the fish behavior, for example. We have people who are doing sort of, Ari, who's actually on this call right now. He's a grad student in the lab, and he did almost entirely computational work and had graduate students and technicians helping him with the zebrafish work. So I think there's a place for everybody, and we really are trying to expand in the area of the tool development and drug discovery. So we just had a preprint from Ari's PhD work where we're working on a new method for drug discovery. And we found lots of molecules that work in zebrafish. So sort of taking it to the next level of having an organism where we really push forward our protein designs or discover drugs and test them in an animal model. And zebrafish are really amazing. They are very small. They fit in 96 well plates. They're entirely see-through, so you can image every cell in the brain, which is why I was drawn to it after my PhD. So projects we're sort of interested in or working on, continuing to work on drug discovery algorithms, particularly drugs that disrupt really specific protein-protein interactions. We're just submitting a grant now. We're looking at an interaction that's critical for long-term memory formation, and we want to see if we can facilitate that to improve memory. Protein binders to manipulate signaling. We've been using Bindcraft quite successfully. New tools, whether genome editing or new tools for neuronal activity, and also collaboration. So we're in a biochemistry department. We have an outstanding cryo-EM core facility here, and PIs that work on cryo-electron tomography, cryo-ET, molecular dynamics. And I'm starting a project with Brian Kelch to look at some of these proteins involved in learning and memory. The big picture, we really want to make progress on these neurodevelopmental disorders, and I think there's a place for some of this, for people who have these backgrounds to be able to do that if you're interested in making sort of a leap to a slightly different field. All right, any questions?
I have a question. How big is your lab currently?
Yes, my lab right now is 10 people. We have 3 postdocs, 4 students, a software engineer, and 2 techs. So pretty decent size. I think I'm going to keep it around that size, somewhere between the 10 to 12 range. But we have a couple of people leaving in the coming years. So far, the postdocs haven't gone. I'm an associate professor, but my postdocs didn't come right away, and so none of them have gone on to new things yet. One of them is really set on academia, which I think is could be a great fit for somebody who wanted to sort of build a program around these 2 different areas. Yeah, and I think anything else?
Do you find that you have postdocs come from more toward the computational side or more towards zebrafish? Because it is really cool that it's this fusion of different areas.
So actually very different. So one of my postdocs is very behavior. So, we use a lot of coding in all of our behavior. I realized after moving from David's lab to this zebrafish lab, how useful all my coding skills were. So animal behavior also involves a lot of that. I have an animal behavior, my software engineer just built a new zebrafish tracking tool that just got a preprint a couple days ago anyway. So it's also very integrated in that side of things. And the image analysis for the neuronal activity, that also is very computationally heavy. But I would say that the people in the lab were mainly doing the Rosetta protein type work. Ari is a PhD student. He started right around COVID, and he was at his house, and he had computational skills, so we went with that. And the other person who's doing a lot of it, actually, he came from a zebrafish lab for his PhD, but he had a lot of computational skills and he wanted to do something a little different. And so he actually went to RosettaCon and has been participating in Rosetta, and he's doing Bindcraft and then tests them in the fish, tests his binders in them. So bringing that together. So a little bit different, but I think it could be a good fit for somebody if they really wanted to do something unique or different.
So I am actually starting with background a little because what I do in terms of the logistics is substantially different than many other professors that you'll be seeing today, now and later. My name is Alican Gulsevin. I got my PhD in chemistry at the University of Florida. I did 4 years of experimental work followed by computational work. And then I moved on to actually Jens Meiler's lab at Vanderbilt University, where I worked for 5 years. And now I worked as an assistant professor at Butler University, Department of Pharmaceutical Sciences. Why are the logistics different? Because Butler University, that you see in here, is in Indianapolis, Indiana. And it combines 2 different characters that you typically see separately in US institutions. Butler itself is a primarily undergraduate institution, which means it does not offer any PhD programs. with the exception of where I work, College of Pharmacy and Health Sciences, where there are several graduate programs, and our doctoral level program is actually a PharmD program. So majority of our research personnel are students who work as part of the PharmD program. Now, this has certain advantages. One of them is going to be that we have enough resources to support research without you having to compete with other people. For example, my cluster has about 450 CPUs, 16 GPU processors, all of them decent hardware. And while it may look pretty modest, because no other person is working as a postdoc in my lab, well, those would be mostly open for use, for your use, with some sharing with the undergraduate students who do the rest of the research in the lab. The other advantage is that because it is not an R1 type institution, we have significant freedom in what we can explore. It does not always have to be a great idea that will take 5 years to discover, but these can be more flexible, short-term projects on more emerging subjects in a results-driven way, so long as they're focused that are publishable, because the shorter duration of postdoc, as I'll be talking about, even with the possibility of extension, will not be sufficient to pursue very in-depth ideas, along with the possibility of remote work within the US. There are some bureaucratic issues depending on the states, but we can talk about that as well. Now, in terms of what I do, there are two main lines of research. What you see here is related to Rosetta method development, where we are trying to leverage the existing membrane frameworks to find ways of accurately placing membrane proteins or membrane binding peptides in membranes using a variety of different methods. It's a very concise project funded by the NSF, and it basically works on development of having one one pipeline where you're just giving your protein to Rosetta without having to align it for some other tool. You will be able to put your peptide or protein in a membrane and move on with all the downstream applications. And that currently is funded until this August with a possibility of extension, but even without an extension, I have the funds to support you at least for the full year, but potentially more. The second part is more vaguely described as investigating ligand-protein interactions, both small molecules and peptides. And the reason for ambiguity is these projects have been so far driven by the interests of the students who want to pursue those directions. Those are metabotropic serotonin receptors, 5-HT2A specifically, along with monoamine transporters, VMAT2, for the treatment of anxiety, substance use disorders, and movement disorders, along with nicotinic receptors, my previous training. But in reality, if you think about what we can do and what I'm interested in doing, I want to pursue a direction in which we don't just use docking and MD and AlphaFold, etc., but something that focuses on using the existing methodologies to create pipelines that answer specific questions related to human health. If we have a number of variants that cause disease, can we find molecules, peptide or small molecule, that can specifically target those to address a specific healthcare problem? Do we have any dysfunctional mutations in any kind of proteins? Can we find ways of stabilizing those mutants to prevent those diseases, or find potentially lead compounds on underexplored areas that is enough interest that we can get funding and or find collaborators. So that is the main idea of what I'm doing. The method development project is what I'm looking for, why I'm looking for a postdoc specifically. But beyond that, if you have any good ideas pertaining to research on specifically membrane proteins, we can also come up with creative ways of you having significant autonomy and exploring those dimensions. If you have any questions, you can take them. Okay.
Maybe just the same question, because I would be interested if I would be applying. How big is your lab? Are there any other postdocs or grad students already in the lab?
No. I have one PharmD student doing research on VMAT at the moment, and another undergrad student doing research on the serotonin receptor project. Along with I guess one difference compared to, again, major universities would be that I am personally engaged in doing research. When I graduated, Jens told me that you will be your best postdoc. Because I am taking his advice on that. That I guess it's a good point though. So there would be a lot of work. So most of the postdoc work would be very much in connection with me. So although there is autonomy in terms of what the postdoc would pursue, I would have pretty direct support in helping guide all those processes. So that is, I think, one advantage. Thank you for the question.
I have a couple of slides. Just to say, my laboratory is split between the United States and Germany. So I have about half of my people in Leipzig and half of my people in Nashville. The big research questions that we are pursuing focus on artificial intelligence in biochemistry. So we are working on AI accelerated quantum mechanics. This is showing you how we in seconds.
You're not sharing at the moment, Jens. We just see you.
Oh, that's even better. Seeing me is super exciting. Okay, so now you possibly see it. So we are developing AI accelerated quantum mechanics methods to drill down on interactions of molecules with proteins. You do know predicting the affinity of a small molecule binding to a protein is a long-standing problem, and we believe that this is a mixture of addressing enthalpy and entropy of the interaction, and for enthalpy we really need to understand in a more fine-grained detail how pi-pi, cation-pi, halogen bonds, how they actually work. And you can't do that if you work off the atomic coordinates as we did for ages. The objective here is to create a scoring function that looks really at the electron distribution. Right? Atoms don't interact. It's the electron cloud. And so quantum chemical calculations to predict this electron cloud are slow, but we believe we can accelerate them with AI. And that's what we are doing here. And here you see a G protein coupled receptor, 40% of our drugs bind there, and you actually see a small molecule binding, and you see how the electron shell changes upon binding to the receptor. And we believe that these minuscule changes are going to be critical for really understanding what the binding affinity is. And I think we really think that it's going to make a difference. We can predict micromolar binders, but we can't distinguish between micromolar and nanomolar binders, and I think this approach is going to get us there. Again, we do this in Nashville at Vanderbilt University. My second slide, we also are building an Institute for Drug Discovery in Leipzig, Germany. We have the idea of this institute is to have computation at the center, and then have all the capabilities to make and test molecules. This includes small molecules, antibodies, vaccines, natural products, looking at protein dynamics, peptides, immune therapeutics. We are building a new building there. We also are building a high-performance computing center dedicated to this task. And we have a National Competence Center of Artificial Intelligence. So it's a quite large enterprise. And before you ask, my lab is about 70 people, counting everybody. The institute, the National Competence Center is another 400 or 500 people or something, so you won't see me a lot, but we have a lot of opportunities for you to explore if you are independent.
I guess, are you recruiting people for Vanderbilt or for Germany or for either?
Both. I am looking for talent worldwide.
I have a question about the research. So we work on like similar stuff. I'm in the Gitter lab at University of Wisconsin-Madison, and work on similar stuff, except we're trying to use like Rosetta, but it's very much more of an engineering project, I'd say, where we're interested in like understanding maybe the interpretations of the AI, but we're really interested in is working on like directed evolution campaigns using our AI models. So do you view your lab more as a engineering-based lab, like focused on maybe producing a really impactful biological result, or as a maybe more physical interpretation-based lab where you're trying to understand I'm not quite sure, I haven't worked with quantum deeply, but understanding the like, like how the quantum
Yeah,
mechanics
I guess I think I get your question. I think the lab is incredibly wide in terms of things of what we're doing and why we're doing those, because it's 70 people. But the overall direction follows what we learned from the Nobel Prize. And what we've learned is that everything that's published right now is based on atomic coordinates. And that is intrinsically limited. It takes the coordinate of the nuclei and trains some AI. We all know that the electrons are actually doing the interaction. So I think one of the next big steps is work towards that. Second, most of these technologies that have been published do ignore protein flexibility. And I do believe that one of the next big pushes in the field needs to be to understand how proteins move and how their movement is critical for function, and specifically how their directed movement is critical for function, which means you want to exclude motions that are not function bearing. And I think this negative design of conformations that are not supporting the function you're looking for is a very big and unaddressed problem. Third, while the latest iterations of the fusion design and AlphaFold prediction feature that they can look at things that are not the canonical 20 amino acids, the prediction quality quickly deteriorates. I think we need to develop AI technologies that are trained with more limited data sets to give us higher accuracy for these kind of situations. I think these are the three big ventures I'm interested in, and these are the three big things we are pursuing.
Yeah, thank you. That gives me a good scope of your guys' work for the next, say, 5 years or something. I don't know if that's what you meant by that, but sure.
I actually think that's a really interesting question, especially like in this space, because it does seem like, yeah, you can use the tools to engineer a specific goal, not necessarily trying to understand how they're working, or you can really try to dissect them. So I'd be curious what Ajasja, Summer, and Alian, how you approach that in your own labs?
I think the things we're trying to understand are not usually the protein level. We're trying to understand, things on the cellular and circuit or like, developmental level, and then it's more of a tool and goal for the protein work.
Yeah, so I think we sort of approach it a little bit more towards probably the engineering side, especially the Walker. So, I mean, we don't dissect, for example, if we're designing some oligomers, we don't spend a lot of times on the failed oligomers, right? Like most people in protein design, you just focus on the ones that work. So, but when we get something that will work, we hope to sort of show why it works and why the mechanism of walking. I mean, if it's gonna walk, it's gonna be pretty obvious from the single molecule tracking trajectories. So, and then with the power source, we'll show that it works by itself. And also, since I'm on the microphone, I had a little bit more time to think about what sets my lab apart. In the uniqueness, and sort of another thing is that we have the single molecule tracking techniques and equipment combined in the same lab with the protein design. So our loop of design test, design build test is easier. It's faster to iterate.
I guess for me, it's in larger labs, you have a scientific question or problem on which you do work to generate some certain type of data, which will create more questions, and you basically keep following that overall trail. For me, again, considering the scale of what I am doing, I am interested in finding the right answer and asking the right question on that, which is to say that there is a lot of data in protein work, structure activity relationships, both for peptides and ligands, in a way that, yes, we know those molecules may work, but we usually do not understand the governing principles behind them. And I believe that especially with the acceleration in AI-based methods, we are getting farther and farther away from understanding the actual reasons as to why things work, and that we focus more on the engineering approach to identify things that do work. And I would say, intellectual interest rests more on, we do have a lot of tools, including AI and traditional methods, that can help us look into existing data and try to understand some of the key principles that may help us understand why things work the way they do. It can be as simple as some of the toy project I worked on. Why do certain probiotic strains work in a scientifically proven way, whereas the others not? And when I explore that, I see that there are certain peptides involved that are also found in foods that have high satiety. What may be their protein targets? Does literature exist on those interactions and sorts? So it's more about connecting the dots between whatever is already published from a computational perspective, more than developing the very tools that will help drive those explorations. So it is a different kind of interest as opposed to many labs who have a lot more resources available to ask and answer those questions standalone.
I guess I have one. Like, how do postdocs tend to like integrate into your departments? Like, I know different institutions kind of have different, organizations, or just like maybe different departments have more postdocs compared to some others. So yeah, maybe a sense of that at each of the places that you're at.
We have pretty good number of postdocs at UMass Medical School. Our department itself has weekly research seminars, which postdocs and students present at. We have lots of other social things like weekly tea time where people meet and socialize. And we have a postdoc union now, it's pretty good situation for them here, I believe. So.
To say I can answer because I have less to say. Postdocs are relatively uncommon at PUIs or I guess, smaller R2s. So from that perspective, it would be fairly unregulated, which means you and I can probably come to an agreement regarding the boundaries. But beyond the funds from the grant that, pay for the main postdoc position, I also do have a decent amount of startup money left that can be used to send you to conferences. New equipment is needed, we can work on that as well. So there is some room for doing something, again, in a way that we can decide as to how things should be working in the absence of any established standards.
So we're pretty well integrated into the department as well at the department level. So we also have weekly department meetings. And so in my lab currently there's only one postdoc, but at the department level there's probably, I don't know, 10 or something. And the other thing is that we don't officially have a postdoc position. You're basically a research position, which you sort of set the boundary for a few years, but there's no limits to how long a postdoc can be. Like, not and other than that, all the other things regarding employments, like pay grades and everything else, is set by the union and other regulation. So this is also pretty well covered and standardized and works well, I think.
Maybe I can chime in and share some more general observations. The Rosetta community as a whole has been a very good training experience for postdoctoral fellows, and we have a good track record of giving, maximizing your potential. There are seed funds to join, and if you join the commons, and there are training opportunities for your graduate students, undergraduate students, and so on. Naturally, a lot of this depends on where you want to go and what you want to do, right? My observation is that over until 2000, let's say 24, everybody I trained as a postdoc was eaten up by industry. Since then, the theme shifted a little bit, and they are looking for people who are trained in computer science and fundamental AI, which a lot of the people who postdoc with me were not. I think this theme going to continue. So the climate going to get a little bit more harsh, but the Rosetta community is a good heaven to endure that. I believe that to move forward in academia, you need to have your very own topic that you own and combine it with AI and machine learning technologies. To move into industry, I would make you take some rigorous courses in artificial intelligence so that we can say you got a training even if you're not a computer science or AI trainee. And everybody of the PIs here will sit down with you and make that plan. Everybody of us sitting here has the capabilities at their university to make that happen. I think we are very much aware about the changing nature. We are moving from, in the next years, we're going to move from more fundamental AI development to application. It's going to take experts in both fields, biochemistry and AI, to really make that work. But as David Baker said, you can watch on YouTube his address to us at the Nobel laureates, there are going to be 10 more Nobel Prizes for applying these technologies that we are developing into biomedical research. And we already, wherever you come from, whatever you want to do, we are ready to train you. We might put you through, if you come from a biochemistry, chemical background, we might have to put you through some very basic science, AI teaching, because it's getting more and more difficult to get positions without that background. But we all believe that there will be opportunities, and we will work with you individually to find the best possible way wherever you go. Right.
In case people didn't see my head nodding, I'll definitely second everything that Ian said.
And I guess what I can add is, having a postdoc research institutions, what Jens has said is true, especially about Vanderbilt. People usually either saw it as a job in industry, which a lot of people did, or a job at an R1 institution where you're doing, kind of research involving postdocs, grad students, etc. But one thing that is not very commonly discussed, especially in larger labs, is that there are other academic careers implementing education, teaching into research positions, either at institutions like Butler, PUIs, or R2 institutions. And unfortunately, the attitude in R1 kind of research can be a little snobbish towards such research, but it is not doing poor quality research. It's just doing a different type of research that still can answer some good questions, but at a smaller scale. But the bottom line is, even in that context where you have a heavier emphasis on education, AI still is a huge thing for many institutions, whether they understand the implications or not. So yes, having some connection to the AI dimension definitely is an advantage, although the meaning can be different based on what kind of an institution you want to go to. That can be, of course, method development on brand new subjects in larger institutions, or implementation of the tools that already do exist to address, again, still relevant questions in smaller institutions. So, but AI be AI, that aspect does not change, and probably will not in the foreseeable future.
Okay, maybe I think we open this up for the last few minutes, if we don't have additional presentations, to the candidates that are here. Is there anything we can answer that would help you to make good decisions for your future?
Maybe like, what criteria did you guys use when you guys made decisions about your future? Like, simple
Oh,
question.
this is super simple. Should be in their 40s or 50s, not 68. Should be working in a field that is super relevant and where there's a lot of potential. And I think that's, for you it's harder to decide, because AI is sort of run its course, and obviously you're going to have a big role, but you have to think about what's the next big step in that area. For me it was easy. In the 80s and 90s nobody was doing AI in a programmed neural network, and I went to David Baker because he was mid-career, super young, had Rosetta, and I could add all of the AI shit to Rosetta that I wanted, and nobody would stop me. But I believe what you have to do is you have to identify one of these areas of development and really dig in. And I mentioned a few of these in my talks, the three big directions I am seeing, but I am I'm a stupid old man, right? I mean, you might see things that I am not seeing, and you might have ideas that I wouldn't know about unless you told me. And this is what I think, what I tell my graduate students: if you approach a PI for a postdoc, come up with your own innovative ideas and tell us about those, because we don't want to do with you the research that's done now. We want to do the research that's hot in 5 years, right? And so I'm not looking for a technician. I'm looking for an original thinker who has their ideas. It was, you know, looking back, it was super easy for me, and I am not super happy for you guys going through this now. But at the time, I had a document. I had a silly text document where I recorded innovative, cool ideas that I encountered as an undergraduate or graduate student, and that I could push if I had the resources and time. And I think I'm speaking for the PIs here. That's what we're looking for. We are not looking to give you a project. We are looking to empower you to take your own ideas to the next level. And so, you know, whatever you guys think is exciting, I want to listen to it. I know some of the things that I think are going to change the world in the next 10 years, but I am super old and I might not have a complete understanding of what will be possible. But as a postdoctoral applicant, tell us what you are about and what you are excited about. That I think is the most important thing, because we don't want technician, we want post.
I think what Jens has said is very insightful, but I would also add that it is in part about acknowledging who you are and what you want. Just because something will be hot in the future, if you genuinely don't like it, don't do it. Like, antibodies are incredibly important, will be incredibly important, but I never had absolutely any interest in working with antibodies. So even if it was $1 million business in the coming 3 years, I wouldn't do it because it's not something that interests me. Find something that you genuinely are interested in, something that you're passionate about in one of those fields. Just because something is the hottest, it doesn't mean you should be doing that very thing in a way that you will not be content with what you're doing. Because what Jens describes requires an incredible amount of intrinsic motivation, that if you genuinely don't want to do something, whatever people around you may push you into doing, work 80 hours, 90 hours, this or that, I don't believe that you will ever be as successful as someone who is doing it because they genuinely want to do it. So think about what really works for you in addition to what will be hot in the next 5-10 years.
I just want to say something briefly because I changed fields so much. I made that decision partly because I knew for sure I wanted to do academia. And when you're thinking about in that way, you want to build your own research program, right? And it needs to be unique. And I thought if I did something quite different, then I can bridge these two areas, and that makes me the only one who can do, that combination. And I think that picking a postdoc with a decent training potential is a good way to go, especially if you want to do academic route. So, for example, if one were to come to a lab like mine where you do a model organism, that, you can write an F32 and that nobody's going to argue that you have a really high training potential there to go and learn a new area. It doesn't have to be as far as model organism, but pick something that is exciting but a little bit different so that you can kind of make your own space.
And maybe lastly, the lab should have a good vibe as well. So you should talk to other people and like see how things are working. Definitely always talk to group members. I visited the Baker Lab twice, and I like did the Baker Lab ping pong when people would like just send me from one person to the other, and that was amazing. And then I just decided, yeah, this is the place I want to go.
Okay, I think we are up on the hour. Are there any additional questions you guys want to ask? Stephanie?
No, yeah, I think this was a really great and fun discussion. Yeah, thanks to
Yeah, and then don't hesitate to contact us. Rosetta community has a really good track record in promoting postdocs. As I said, I do believe that you have to have your own unique ideas of what you want to do and how it's new. But we are ready to receive you and work with you on developing those ideas and moving them forward. Thanks so much for Camille, Julia, for organizing this. I'm not sure if any other organizers are here, but don't hesitate to contact us or any other PI in the Rosetta community. You'll find a complete list on the Rosetta Commons. We do have a track record and we are ready to work with you. I think we just try to illustrate where the future is going and what the opportunities might be. And we have representation on all across the entire world. America, North, South America, Europe, Asia. We can put you in contact with the right people.
Yeah, and also Stephanie, thanks for organizing, or at least you sent the emails, right?
Oh yeah. Thanks. Yeah, if also I wanted to say, like, if people have questions, like, I was a postdoc who's not on the Rosetta community before my postdoc, so if you want to talk to other postdocs, feel free to email me, or like Ajasja said, I think it's always great to reach out to people in the labs that you're thinking about too. I did that a lot, and that was very helpful in my postdoc search. So yeah, but with that, thanks everyone.